Hey everybody, this is group one, and this is our module 3 project for PyMOL. And in this exercise, we were investigating beta glucuronidase and E. coli. And group one was myself, Lainey, Martin, and Becky. So beta glucuronidase, more formally known as GUS, the reaction for this enzyme is that the enzymatic hydrolysis of beta glucuronides from glycosaminoglycans and urine and other fluids. And over on the right there, you can see a picture of our enzyme. And our enzyme GUS is usually located in the lysosomes primarily, but it's also found in the convoluted tubules of kidneys and also the epithelial tissues of kidneys. Some classification of our enzyme is how it's a homotetramer that's made of 4 identical subunits. And some of the structural configurations shown in our image on the right include alpha helices, which are shown in green, and then beta sheets, which are shown in the blue arrows. And also our enzyme contains a jelly roll barrel and a tin barrel as well. The significance of our enzyme is how it's involved in the breakdown of, as I mentioned earlier, beta glucuronides. And this breakdown allows for reutilization of the products as glucuronic acid is used to neutralize harmful compounds and is excreted in our urine. It's also used as an energy source for some forms of enteric bacteria and how it breaks down the glucuronic acid into an energy source. Some research going on with our enzyme is how there's side effects from cancer chemotherapy drug CPT11. This drug is also known as irinotecan and is activated and then converted to inactive SN38, which is then sent down to the GI tract. Here, glucuronic acid is removed by GUS, which consequently reactivates the SM38 into a toxin that causes damage to the epithelial lining and causes diarrhea. Next, we're going to talk about some of the key features of our enzyme, beta glucuronidase.
The first key feature we're going to talk about is the active site of the enzyme. The active site is part of the enzyme that directly binds to the substrate and carries a reaction. It contains the catalytic groups glutamic acid, aspartic acid, valine, phenylalanine, tyrosine, and arginine that promotes the formation of hydrogen bonds with the inhibitors. The picture to the right shows these residues and how they interact with the inhibitor used in the most recent study, with glutamic acid 413 being the closest to the inhibitor.
The next key feature we'll talk about is the bacterial loop in beta glucuronidase. It tends to fold over the active site of microbial enzymes. It is only present in the bacterial samples and not in mammals. Inhibitor binds to the bacterial loop, causing them not to be effective against mammalian beta glucuronidases. Where the inhibitor's binding determines the activity of the molecule, the initial contact is located at leucine 365. This is where the inhibitor binds through hydrogen bonding. The full loop is located at leucine 360 to serine 376, shown in dark purple in the image. The activity and function of the protein is determined by the inhibition of the bacterial loop. When there is an increase in inhibitor activity at the bacterial loop, it decreases the activity of the enzyme. We will now talk about the reaction mechanism of GUS.
So for the reaction mechanism of GUS, on the right, we have a picture of the active site showing the 6 active site residues: glutamic acid, asparagine, valine, phenylalanine, tyrosine, and arginine. The inhibitor performs hydrogen bonding with the glutamic acid 413, which is the closest to the inhibitor. The amino acid residues are shown in blue, while the inhibitor is shown in yellow. And once the inhibitor binds the active site, it also has hydrophobic contacts with leucine 361. And it deactivates the bacterial loop, which in turn deactivates the entire enzyme of GUS.
So for GUS in module 4, students will be expected to understand the enzymatic mechanisms and the hydrolysis of glucuronic acid from glycosaminoglycans. And we'll be doing this by observing the changes in absorbance per minute compared to the concentration of GUS. After we figure out the concentration of GUS used for its highest enzyme activity, we will then use this information to find the optimal pH range and spectra of light for that same highest enzyme activity.
So there were two key features that we discovered using PyMOL. One of them was how to make the background pieces of the enzyme translucent so that we could really focus in on the key features that we were looking for, like earlier with the bacterial loop. And then the other thing that is kind of hand in hand with that is how to make specific objects within the enzyme so that certain things could be different colors from each other to again show the pieces that you really want to be talking about.
We chose our pictures because they displayed the key features that showed major interactions which could lead to function or to cease a function, such as interaction between inhibitor 8 and the bacterial loop, along with the active sites where it binds in the overall enzyme as a whole.
It is important to provide good visualization and rendering skills because it provides clarification for research papers and allows for scientists to easily locate and identify the key features for further analysis.
Thank you, guys. Okay.